Our first presenter will be Gabriela Canas. Gabby is a second-year master's student in the University of North Florida's Coastal and Marine Biology flagship program, studying estuarine ecology under the advisement of Dr. Nikki Dix. She is currently a Dolores Ozine Fellow. Her thesis is focused on the effects of climate change and shifting vegetation range on benthic algal communities. She serves as the field technician for the Wet Feet project, where she is actively involved in on-site research as well as the development of virtual reality-based education content to communicate project topics. So Gabby, I will hand it off to you.
Gabby, it looks like you are muted still. Thank you.
Thank you so much, Abby, for that awesome introduction. So I'd like to just start by saying how immensely grateful and blessed I feel to be a part of the amazing community here at the GTN. These photos don't even begin to cover all of the incredible researchers, educators, CTP administrators, everyone, volunteers, everyone I've worked with and had the opportunity to quite literally live out a childhood dream. So my love for coastal systems and marine science started very young through various fishing trips with my family and a lot of other nautical ventures, but I learned from a very early age and developed a passion for coastal systems. And I've been so fortunate to be able to put that passion into practice through my work with the Wet Feet team. They're doing so many amazing things, and my small little piece of that project is basically I'm the person who goes out and wrangles the chambers when they decide to strip and run around the marsh. So I've had a lot of time to do some heavy lifting and get up close and personal with these chambers, but I've also had a lot of time to get up close and personal with Florida's beautiful coastal marshes. And that's afforded me the opportunity to learn a lot more about the questions that the Wet Feet project is asking about soil elevation change, ecosystem carbon gain, all of these questions associated with the shift from salt marsh dominated to mangrove dominated systems. But it's also given me some time to come up with some questions of my own. And that was the basis of my master's thesis, which is asking, the question of how will changes in temperature and the accompanying changes in foundation species affect community dynamics? So as someone who grew up loving marine organisms, that's where my mind went when I heard about this shift. And as you can imagine, this is a very broad question that is not answerable by one person, I was advised by some very awesome people to start small, and I took that advice quite literally, and I started microscopic and at the bottom. So what exactly is the bottom of the food web in these systems? Well, that is a topic that's currently up for debate, but a lot of recent research is suggesting that a large portion of the primary production energy for the food webs is actually coming from benthic microalgae. So stable isotope analysis is letting us look at these systems, and basically it's the idea that you are what you eat. And in this graph right here, we see BMA, that benthic microalgae, in the dark gray, and a lot of organisms are falling out right above that benthic microalgae signature, suggesting that they're heavily relying on those organisms as their primary source of carbon. Fresh, hot off the press, some studies in Louisiana and here in Florida are also suggesting that these microorganisms are likely contributing more primary production to food webs than even the salt marsh or mangroves themselves. So these are very important organisms. When I say benthic microalgae, what am I referring to? Well, I'm talking about the small microscopic unicellular organisms that are inhabiting the upper several millimeters of illuminated sediments. So these guys can be further divided into three different subgroups. You have your blue-green algae, your cyanobacteria. you have green algae, and you have diatoms. And these groups are extremely diverse, and they all respond to system change differently. And they have various implications for our system. So blue-green algae cyanobacteria can often form harmful algal blooms and release toxins. And things like green algae and diatoms are that readily consumable labile carbon that a lot of these things, these grazers, are foraging for and actively seeking to consume. So how could potential changes in vegetation affect our benthic algal community? So at this point, you guys should be familiar with a lot of these plant species. We have our black mangroves, our Batis maritima, that's succulent, and we have Spartina alterniflora, our primary marsh grass. And these are the major heavy hitters in our marshes here in the GTM Research Reserve. And they have the potential to affect the benthic algal community in a wide variety of ways. First off, their structure, their canopies, can impact the amount of light that's reaching these soils for these photosynthesizing organisms. Not only that, but light and shading can impact the temperature of the soils. And like I said earlier, these different groups respond differently to different temperature ranges and grow optimally in different temperatures. These plants also have below ground structure, which kind of sets up the habitat and the availability of access to these sediments for grazers. So the root systems of these plants can be very important too for structuring these systems. Roots, and as the wet feet team is studying, the way these plants grow can also impact the elevation at which they're found. And elevation plays a big role in tidal inundation. So as water comes up over the marsh, that is a source of resuspension for these algae. And the structure of these plants can control how fast the water is flowing through these systems and potentially how much of that algal biomass is getting resuspended up into the water column. So all of these potentials leave us with a lot of questions as to how the algal community will actually be affected by these different vegetation types. And that's where I come in. So the questions that I'm particularly interested in are, how does the benthic algal community differ under different vegetation types? And also, how does increased air temperature affect the benthic algal community? So I have the amazing infrastructure of the wet feet team, and also all of these changes that we're seeing are driven by climate change and increases in temperature, decreases in freeze events. So it's important to know how that factor in particular can affect these communities. Today, I will be talking to you guys about my first question, that vegetation question. And I've been fortunate enough to have the existing infrastructure of the wet feet team to choose my pick of the litter of the marshes here in the GTM to conduct my study. So I chose the middle of the 3 wet feet sites for my study. And it was kind of the Goldilocks marsh for me because it has monocultures of all 3 of those primary vegetation types. It has patches of Batis, patches of Spartina, and patches of mangroves. And I was able to choose 3 different 100-meter squared locations within a single creek system to study the different vegetation types. So again, my 3 treatments were Avicennia germinans, those black mangroves, Batis maritima, that succulent, and Spartina alterniflora, our smooth cord grass. My project is a bit complex and has a lot of different moving pieces. I'm studying not only vegetation and in situ site parameters to establish how those different vegetation types are composed, but also warming as well. And then I collect sediment cores to take back into the lab to analyze the benthic algal community in a few different ways. So I use chlorophyll analysis to look at overall biomass. I'm also currently going through samples of mud to identify species composition and community composition using microscopy. And we also have samples preserved for DNA metabarcoding, which is a newer novel technique to also get at that species composition, that community composition, which is so important. Today, I'll be focusing on the in situ vegetation parameters, as well as the extracted chlorophyll biomass parameters. So what are some of the things that we analyzed? Well, we looked at temperature and salinity. Temperature, we used hobo data loggers to get Bluetooth temperature readings. And then we also had to get a little bit creative to get salinity. It's kind of like getting water from a stone if you've ever been out in these marshes. We scooped mud into a syringe and squeezed water out of the mud onto a refractometer. So that was a fun measurement for sure. We also used photosynthetically active radiation. So this is the visible light spectrum which these photosynthesizing organisms are actually utilizing. And we get above the canopy readings and below the canopy readings and report that as a percentage of the amount of light that's reaching the sediment surface. We also, thank you to Ches and other wet feet team members, got really accurate millimeter accuracy elevations, which gives us an idea of the elevation in these different vegetation types. And then we also, thank you to Christine Angelini, got biomass in situ, which not only gives us the biomass on site, but can also segregate out how much of that biomass is contributed by the 3 different algal groups. So diatoms, green algae, and cyanobacteria, all in one instantaneous reading. We also use those sediment cores to get extracted chlorophyll A, that biomass proxy, using pigments and fluorescence. And what did we find? So our first question of canopy influence on light, we found that there were differences in light in the 3 different vegetation types. So Avicennia had significantly lower levels of light. Batis was fairly intermediate, with Spartina falling out with the highest light levels overall. For that temperature question, we saw that there were also differences in temperature. Again, we saw that kind of stair-step pattern with Avicennia having slightly lower temperatures, Batis and Spartina falling out with slightly higher temperatures. For elevation, thanks to Ches, we were able to get some interesting elevation measurements, and we found that Avicennia and Spartina generally have similar elevations, and Batis is falling out slightly higher in our marsh platform. For salinity, we saw not really very many differences. The salinity was kind of all over the place and likely not controlled by vegetation. But what about the star of the show, our algae? Well, we had some interesting findings. So first off, this is our first sampling event in the spring, which is our peak algal biomass. And I'd like to draw your attention, this is a lot for a graph, I know, but I'd like to draw your attention to this group here in light blue. These are our diatoms, and they're making up the majority of the biomass in our marshes, which is to be expected. Coastal systems are often diatom dominated. We saw in situ that Avicennia and Spartina had fairly similar levels, with Batis falling out slightly higher. And then we also saw that cyanobacteria seemed to be slightly higher in the Spartina dominated areas. In the summer, we saw overall significantly lower levels of chlorophyll, of biomass. Again, we saw that kind of diatom dominated system, and we saw almost a reversal of the in situ patterns that we saw in the spring. So here we see Batis being the highest and Avicennia and Spartina falling out lower, and Batis also seem to have slightly higher levels of cyanobacteria. The story really starts to get interesting when we look at our extracted chlorophyll levels. So these are the slightly more accurate measurements that we do in the lab. And here we saw that stair-step pattern that should look kind of familiar to earlier. We see Avicennia with the lowest amount of active chlorophyll here in light blue, and Batis and Spartina having slightly higher levels of active chlorophyll, with Batis being that intermediate. This dark blue measurement is phaeophyton, which is degraded chlorophyll. So this is decaying algal biomass that's in the system, and we saw fairly consistent levels across all 3 treatments. And then when we go to the summer, we saw overall lower biomass in the summer months, but we did see, again, lower levels in Abyssinia and Spartina and Baetis slightly higher, with Baetis being the highest levels in the summer, which was interesting. So how do our algal parameters relate to some of those conditions that we saw? So this is a lot, so just to orient you, we have our algal biomass extracted chlorophyll measurement on the y-axis with temperature on the x. And some of the trends that we saw was that as temperature increased, we also saw a corresponding increase in algal biomass, which was significant. Light as well showed us a generally positive trend. Salinity was, as you can see, all over the place, so not really any trend to dissect there. But elevation, although it wasn't statistically significant, did show a general positive trend with our algal biomass. So some interesting findings. And although this opened up a lot more questions than it answered potentially, we did have some findings. So we know that conditions between different vegetation types are different, the abiotic conditions. We also know that algal biomass is different under different vegetation types. And we are starting to understand that conditions like temperature, elevation, and light are likely part of the story that's driving the changes in algal biomass. We still don't know how the algal community composition, that species composition, is potentially changing. We're trying to find that out actively. Shout out to Paige Priester. We also are trying to understand how the algal community composition is changing under warming conditions. That's the next part of my project. Stay tuned. And finally, we need to get a better understanding going forward as a research community of how changes in benthic algae will affect food web dynamics in these marshes as a whole. We need to go back to the basics. It seems like algae, benthic algae in particular, is kind of lost popularity. And we need to bring those back because it's looking like they're really big players in these marsh systems. All right, so I'd like to acknowledge all these people and questions. Ooh. Sorry.
All right, thank you, Gabby. I don't see any chat box questions quite yet. Anyone still typing? Okay, I see a question. As far as community composition goes, are you looking at the presence or absence, or are you trying to quantify density?
Great question. All of the above, actually. So with community composition and specifically the cell counts and microscopy, we are able to get an idea of species presence through identification, not always down to the species level, kind of higher, lower resolution, I suppose. But we can also get an idea of density because we know the amount of soil that we're counting and the cells present in those soils. So all of the above. Great question.
Thank you. Well, in the interest of time, we are going to move on to our next presenter, but Gabby, if you are able to stick around in the chat box and answer any further questions. Thank you.